Thank you very much. Thank you for the invite. This is a really exciting time in DNA writing, and so I'm really happy that I get to share this with new people. Okay, so I just want to start by thinking about what are the three primary information systems that we humans use to encode ideas and function, and their language, software, and DNA. And DNA in some ways is the oldest because it predates humans, but it's also the youngest, and it's the youngest information system that we've been able to master, and we're still in the process of mastering writing and programming in it. And so these systems have something in common, which is that they all encode instructions. They're all interpretable by machinery, your brains, your CPUs, or your cells. They can recursively build, evolve, and bootstrap new systems. And most importantly, they require tools to manipulate. And I think it's important to think about, the tools really control what you can do with these systems. And so it's important to think about, when we're thinking about the tools to manipulate DNA, to think about the analogies to language and software and how we can, where we are in that process. And so I would just like to put forward the idea that we're basically had three eras of DNA engineering that each lasted about 20 years. We've had the collage era, where we could just cut and paste DNA, and that's it. We had something like the printing press era, where we could write a little bit of DNA and we could make copies of it. We had the punch card computer era, which is either still going on or just ending now, where we could write a little bit better and then make very simple programs with a lot of effort. And hopefully we're entering the modern computer era, where we can write long pieces of DNA and also program. And so I'm going to walk through the technologies that led to each of these eras and then what we could do within there. So let's start with the collage era. There's really just two main technologies in this time span, and that is restriction enzymes and ligases. And restriction enzymes are part of the immune system of bacteria that cut at very specific sequences of DNA, usually 6 bases or 8 bases, and to keep out foreign DNA. And they leave these overhangs. Let's see if I have it. Yeah, so they leave these overhangs. And these overhangs can be a substrate for ligases, which bring them together. And with these two functions, we can cut DNA and we can paste it together, and that led to recombinant DNA, which is basically two pieces of DNA that aren't natively together. And we could use recombinant DNA to learn a lot about biology. The analogy here is that we have ancient texts in a different language, and we have a scissors and a piece of tape. And with those tools, even with those tools, we could characterize genes, we could create transgenic genes, and in fact, make bioproducts like insulin, and it was really the foundation of molecular biology. But moving forward, we really needed to write DNA. And that came about in the early 1980s. A reliable way to write DNA is phosphoramidite DNA synthesis. And this is just a chemical process that you can go through in cycles to add a base successively to create an oligonucleotide. In the 1980s, we could do that about for 20 to 30 bases, and then the rest of the time we've just been learning to do this at a higher efficiency and fidelity to get it longer and longer. And this is still the dominant technology we use today to write DNA. Today we're at about 200 to 300 bases, and it's miniaturized and a very mature technology, and it's unlikely to go much longer than 200 or 300 bases. Just the error rates are not coming down. So this is really the maximum by which we can write. With the ability to write, we also gain the ability to copy. And so here what we're doing is we're taking these small oligonucleotides and using them in a polymerase chain reaction. And so here we're just taking small nucleotides, annealing them to existing DNA, and then extending with a polymerase, and we can make many, many copies of the cell. And this technique was invented in 1983. It got a lot better when we didn't, with a thermostable attack polymerase, because this denaturing step here would kill the polymerase every time. When we found one that could survive that, it got a lot higher throughput. And this is a very early PCR machine, which was just three buckets of water, essentially, that you would move a tube between the water. Today, we have modern PCR machines that cycle between the temperatures. PCR is still a dominant technology today, and but it has its caveats. You can get to about 5 kb of copying, but after that, it gets quite hard. And there's a lot of DNA that's hard to PCR, high GC content, and repeated DNA. The last technology in this printing press era is the DNA editor. So this is the idea that with these new pieces of DNA that you could write, you can introduce a mutation and then PCR, and that mutation gets incorporated into your new DNA. This is a site-directed. These have all won Nobel Prizes, including this. So this is a site-directed mutagenesis. So at the end of this era, we're at the beginning of the 2000s, we could write short oligonucleotides, we could copy gene-length things, and we could edit nucleotides. That allowed us to clone, amplify, and modify any gene from any organism. It was the beginning of structure functional analysis because we could make precise mutations in a protein and see its effect. And it really transformed molecular biology from observational to mechanistic. But it was a very manual and incremental process. And we lacked the real ability to do any programming, right? We couldn't write DNA from scratch beyond these oligonucleotides, which were just not quite genes. So that changed really in the late 1990s and the 2000s by this technology called polymerase cycling assembly. And so this is a lot less known of a technology, but essentially it's just printing a number of oligonucleotides that have partial homology to each other, then extending with a polymerase, and then sealing the gaps with a ligase. And this really transformed our ability to write longer pieces of DNA, gene-length pieces of DNA. And when you buy a DNA gene today, it's most likely created by this method. So people are using phosphoramidite chemistry to build out these oligonucleotides, and then they're extending it. And there has also been a race to make this able to accommodate harder in sequences, high GC content and repeats, and to have it go longer. One of the big breakthroughs is that you could pair it with an error correction mechanism to extend the lengths. And today we're at about 2 to 3 kb. But because it's a PCR-based method, it has a lot of the same limitations as PCR. There's a lot of DNA that's inaccessible. So to go longer than 2 or 3 kb, you really need something that's called assembly. So in modern day, we call everything before the synthesis up to PCA, and then assembly is anything longer than that. And assembly, I showed you an assembly method, which was restriction enzymes, but that's very limited because you had to find these sequences that you could cut. Gibson assembly changed that. And so what you could do with Gibson assembly is you could PCR 2 pieces of DNA with homologous and create homologous ends. Then you use an exonuclease to cut back the ends, a polymerase to fill them in, and a ligase to seal those ends. And that effectively lets you join any 2 pieces of DNA without worrying about the sequence. So it's a sequence-free DNA. So this was developed in 2009 by Daniel Gibson. It's known as Gibson assembly, and really transformed our ability to manipulate DNA, to put pieces together. Is the beginning of sort of programming of longer DNA. So if PCA gets you genes, this gets you pathways. However, it's also limited because it relies on PCR. So again, high GC and repeats are challenging, and getting longer than 10 kb is also challenging. It's not impossible, but it's challenging. The second major technology which allowed you to assemble that we're still using today is called Golden Gate assembly. And so what this did is it uses restriction enzymes, but these are special restriction enzymes called type 2S restriction enzymes, which don't actually cut at the sequence they recognize. They cut distant from the sequence they recognize at any bases in that sequence. And so because they cut there, you can program in the sticky ends. So you can use whatever sticky end you want here and just use the same restriction enzyme. And what that lets you do is essentially a multi-fragment one-pot assembly. So you can create a set of sticky ends and then ligate them all into a plasmid. And this really made the process of assembling DNA modular, which is an important feature of programming. So in modern synthetic biology labs and companies, you sort of have these parts that have these overhangs that are very fixed, and you can put these parts into transcriptional units, and then put these transcriptional units into a multi-transcriptional unit or multi-cassette. And you see here, this is sort of the beginning of reusing DNA, which is a very important feature of programming. However, it's quite low throughput. So you have to just to make this, you have to go through several buildings you have to build plasmids several times, each one with a long process and a lot of hands-on time. And then it's quite rigid. If I want to switch the orientations of these, I have to do a lot of extra work. Although you can program, it's a little bit limited, right? Okay, so this is what I would say is where we are today. So we're sort of in the punch card computing era. So this is an old photo of old computer engineers working with these big stacks of punch cards to write their programs. And that's probably simpler than what we're doing today in biology. But we are able to write, cut, and edit. We can do rudimentary low throughput programming. Maybe someone who's very skilled can write about 100 designs, can get through about 100 designs a month, but that's about it. It's modular, but quite hardwired code. And many sequences are inaccessible to synthesis or assembly. And because of all these problems with assembling DNA, commercial DNA suppliers really don't do much of it. So they will only get you about 5 KB. They'll generally want to stop at PCA or real near PCA. So I want to look towards the future and think about what we need to get better writing and more programmable DNA. And so how do we get from punch cards to something we're all familiar with when dealing with code or language, a code editor that has like all of these functions, copy, paste, spell check, undo, these types of functions that we're all used to. How do we do that with DNA? And I would say it comes down to 4 basic factors. Completeness, so we want to be able to write whatever we want, not limited by sequence. Processivity, it's too damn hard to make a lot of DNA sequences. We need to get faster and easier at doing it. Reusability, I want to share my code with you and have you reuse it. And composability, blocks of DNA code should be able to be cut and paste and rearranged without, they shouldn't be hardwired into the DNA. So I'm going to just talk about a few technologies that may be getting us towards this future, or 2 technologies basically. So one is enzymatic DNA synthesis. This is the first sort of alternative to phosphoramidite chemistry in terms of building DNA. And it's a much simpler process. What you're doing is you're reprogramming a templateless DNA polymerase like terminal deoxynucleotide transferase. So this is the enzyme that makes your telomeres, but there are alternatives. There are other enzymes that have been built, that this has been built with. And all you do is extend and deblock and extend and deblock, and it's a much simpler process. And because of that, it has less errors, so you can get to these longer oligonucleotide lengths. These longer oligonucleotide lengths when combined with PCA, give you more complex DNA. So now we're able to write much more complex DNA. And this is commercially available today. ANSA is an example of a company that's providing it. However, it's a higher cost process. The inputs are quite costly, and there's a lot of uncertainty about whether it's a viable business model because of that. People want things cheap. So I would say the second, and this is one where I'm very biased on, perhaps technology that's leading us towards the modern computer era, is in vivo DNA assembly. So one of the problems with DNA assembly is you have to go through this process of purifying DNA from the cells, doing an in vitro manipulation like Gibson assembly or Golden Gate assembly, joining them like with a ligase, and then putting it back in the cells. And that's just too complicated to scale or do cheaply. It's a hard process. And so what if we can replace that with a bacterial conjugation and a program inside of the bacteria that does this cutting and pasting? Then we could scale. It's a much simpler process. And so that's what we're trying to do at BacStitch. So BacStitch is using a genetic program that combines bacterial conjugation, endonuclease cuts, and homologous recombination. And because you never leave the cells, you also don't have a lot of the problems with long DNA. Long DNA is mostly problematic when you're outside of the cells. So we've been able to reach 100 KB assemblies, and we've been automating it, and it's quite low cost compared. It comes down to, instead of that complicated process, we're going through many cycles. We have one step where we put the DNA in the cells, and then what we're doing every step is we're just pinning arrays of bacteria, and the bacteria are doing the joining of the DNA. We also have some tricks to enable us to sequence in pools, so it's a cheaper spell check as well on this. And it turns out you can do all sorts of things with this. You can subclone, you can edit, you can shuffle, you can deliver it to other cells, you can build combinatorial libraries. And because it relies on homologous recombination, it doesn't have a lot of the limitations in terms of composability that these in vitro methods have. So you can do modular assembly, modular shuffling, scarless assembly, truncations, extensions, fusions, insertions, all without leaving the cells. And so it uses that same process to do all of these processes. Are we entering the modern computer era? I would like to leave it open to the audience, what else we need, but I would say we're getting better at writing pathways, and we're starting to get to a software version of programmable DNA where we can actually manipulate the DNA easily. And enzymatic synthesis is getting to this complex DNA, and in vivo DNA assembly is getting us to this more programmable set of DNA. And this is work that's supported by the SBIR from the NHGRI. We're very thankful for it, and I thank you for your attention.
Thanks, Sasha. Questions? Please use the microphones. You mentioned the delivery of DNA. How do you, where does it go? What's the end point? What's the end product of a big piece of DNA, and how can you use that in an experiment?
Yeah, so I think there's a lot of challenges around delivery. One is just how do you get it into the cell, and the other is how do you get it into the genome, or if it's a mammalian cell in particular. And it's highly dependent on the cell type in terms of delivery. So there's a lot of prokaryotic organisms where you can actually just conjugate between E. coli and that organism. So this platform that we're developing would work. But for mammalian cells, it's a lot harder, and you need to use viruses. And there's a lot of challenges around not just, so if I could write long DNA, can I get it into a mammalian cell? That's a challenging problem that we're not doing, but you know, there is work on like reprogramming HSV viruses to deliver these long packages to mammalian cells, but it has to happen at the same time.
Is there a size limit to the modules that you are finding that can be input into the bacteria reliably and then manipulated?
So right now our size limit is about 10 KB for a block that we can assemble, but we're actively trying to push that, and I don't think we've reached sort of limit on that.